Hi, my name is Erin, and we're going to be discussing ion channels and neuronal communication. As we begin, let's take a look at this diagram of the neuron displayed on this slide. It represents one of the most specialized and complex cells in the human body. Neurons serve as the wiring system that allows communication across the brain, spinal cord, and peripheral nerves. Every movement, thought, and sensation depends on how these neurons send and receive information. This communication occurs through both electrical and chemical signals. Electrical impulses, known as action potentials, and chemical messengers, called neurotransmitters, allow for the rapid exchange of information across vast neural networks. The critical structures that make this communication possible are ion channels, tiny, specialized proteins embedded in the neuronal membrane that regulate the flow of ions like sodium, potassium, calcium, and chloride. These charged particles move in and out of the neuron through specific channels, creating the voltage changes necessary for signal transmission. In many ways, you can think of ion channels as the gates that control electrical current in the power grid. Now, which organ system relies most heavily on electrical signaling? The answer, of course, is the nervous system. While the cardiovascular and muscular systems also depend on electrical impulses, the nervous system uses them as its exclusive means of communication. Without these precise voltage changes, the body's sensory, motor, and cognitive processes would fail to function properly. As we move forward, keep in mind that every neural process, from a simple reflex to a complex emotional response, depends on the orchestrated behavior of these ion channels. Here we focus on the architecture and operation of ion channels. These channels span the neuronal membrane and form highly selective pores that allow only specific ions to pass. Their structural design is a perfect example of molecular precision. Each channel contains a selectivity filter that discriminates among ions based on size, charge, and hydration energy. For instance, potassium channels allow potassium ions to flow while excluding smaller sodium ions due to subtle atomic level differences. So let's break down the four major types listed on here. Voltage-gated channels open or close in response to changes in the membrane potential. Ligand-gated channels respond to specific neurotransmitters like acetylcholine or GABA. Mechanically gated channels open when the membrane is stretched or compressed, which is critical in touch and hearing. Finally, weak channels remain partially open to maintain the resting potential. Ion channels are not static structures. They undergo conformational changes that determine their open or closed states. These movements occur within milliseconds, allowing neurons to respond almost instantaneously to stimuli. There's a diversity of these ion channels, each with unique gating mechanisms and kinetics, which underlies the nervous system's flexibility in processing different kinds of information. So here we show how their activity establishes the neuron's resting membrane potential, the baseline voltage across the cell membrane. As seen in the animation, neurons maintain a steady potential around -70 millivolts. This is achieved primarily through the sodium-potassium pump, which actively transports 3 sodium ions out and 2 potassium ions in for each ATP molecule consumed. The key concept here is that this pump creates both a concentration gradient and an electrical gradient. Potassium ions tend to diffuse out through potassium leak channels, leaving behind negatively charged proteins inside the cell. This imbalance of charge results in a negative resting potential. It's a state of dynamic equilibrium, a balance between opposing forces that sets the stage for rapid depolarization when the neuron fires. To help visualize this, think of the neuron like a loaded battery. The pump charges the neuron by separating ions across the membrane, storing potential energy that can be quickly released during an action potential. If this resting potential were not maintained, neurons would be unable to transmit signals effectively. Action potential. An action potential begins when the stimulus depolarizes the neuron's membrane potential to about -55 millivolts, the threshold value. Once this happens, voltage-gated sodium channels open, allowing sodium ions to rush in. The influx of this positive charge rapidly depolarizes the cell, briefly reversing the polarity of the membrane. Next, as sodium channels inactivate, voltage-gated potassium channels open, allowing potassium to exit the cell. This outflow restores the negative internal environment, a process known as repolarization. However, because potassium channels close slowly, the membrane becomes slightly more negative than its resting state, producing the hyperpolarization phase. If you look at the curve on this slide, each stage corresponds directly to these ion movements. The cycle occurs in just a few milliseconds, yet allows neurons to transmit information at remarkable speeds. This precisely timed opening and closing is the molecular choreography that underlies all electrical signaling in the nervous system. Without it, coordinated thought and movement would be impossible. Here we see how the action potential travels along the axon. Once generated, the depolarization at one section of the axon triggers voltage-gated channels and adjacent sections to open, creating a domino effect of electrical activity. The signal does not weaken as it moves. It's regenerated at each segment, ensuring long-distance communication within the nervous system. The myelin sheath, as shown in the animation, acts like insulation around an electrical wire. It prevents current loss and dramatically increases the speed of transmission. The exposed gaps between the myelinated segments, nodes of Ranvier, are packed with sodium channels. The electrical impulse jumps from node to node, a process called saltatory conduction. This leapfrogging of the signal can make conduction speeds up to 100 times faster than in unmyelinated axons. Diseases such as multiple sclerosis result from demyelination. The damaged myelin slows or blocks conduction, leading to muscle weakness and impaired coordination. This slide marks the transition from electrical to chemical signaling. As the action potential reaches the axon terminal, voltage-gated calcium channels open, allowing calcium to enter the neuron. This influx is created because it triggers vesicles loaded with neurotransmitters to fuse with the presynaptic membrane using snare proteins. Once released, neurotransmitters diffuse across the synaptic cleft and bind to receptors on the postsynaptic neuron. This can either excite or inhibit the next cell, depending on the type of receptor activated. The precision of this process is extraordinary. Neurotransmitter release occurs within milliseconds and is tightly controlled by calcium concentration. Many medications and toxins target these mechanisms. For example, botulinum toxin blocks acetylcholine release, leading to muscle paralysis. This shows how delicate and essential calcium-regulated synaptic transmission is to proper physiological function. Here we focus on ligand-gated ion channels, proteins that convert chemical messages into electrical responses. When acetylcholine binds to the receptor, the channel opens to allow sodium ions to enter and potassium to leave the cell, producing a small depolarization called an excitatory postsynaptic potential, or EPSP. This mechanism is the key to communicate with the neuromuscular junction. It's how a neuron signal becomes a muscle contraction. Ligand-gated channels are also found throughout the central nervous system, playing roles in attention, learning, and memory. Clinically, these receptors are important drug targets. Nicotine mimics acetylcholine, overstimulating the receptor, while certain anesthetics and muscle relaxants block it. Here we examine inhibition, an essential counterpart to excitation. The diagram compares excitatory synapses, which allow sodium influx, with inhibitory ones, where chloride ions enter through GABA or glycine receptor channels. The influx of chlorine makes the neuron more negative, moving it further from that threshold and reducing the likelihood of firing at action potential. This process, called hyperpolarization, maintains stability in neural networks and prevents overexcitation. It's a crucial safeguard. Without inhibition, the brain could enter uncontrolled electrical activity such as seizures. Many sedatives and anxiolytics, including benzodiazepines, working by enhancing GABA activity, thereby increasing inhibition and calming neural circuits. This highlights how excitation and inhibition operate in tandem to regulate the brain function. The fine-tuned balance between these two allows complex behaviors and cognition to emerge from billions of neurons communicating harmoniously. Here we connect the molecular biology to human disease by focusing on channelopathies, disorders arising from ion channel mutations or dysfunction. Examples include epilepsy, caused by sodium channel defects, cystic fibrosis, resulting from mutations in the CFTR chloride channel, and myotonia congenita, due to chloride channels abnormalities in skeletal muscle. For instance, in epilepsy, a single point mutation can prevent sodium channels from closing properly, causing neurons to fire uncontrollably. Similarly, in cystic fibrosis, chloride transport failure leads to dehydrated mucus and chronic lung infections. So how does a single channel mutation lead to complex disease symptoms? Even a single amino acid substitution can alter the conformation or gating kinetics of an ion channel, which in turn disrupts ion gradients and electrical balance. This molecular disturbance cascades into organ-level dysfunction. These examples underscore the clinical importance of ion channels, not just as cellular components, but as vital therapeutic targets in neurology and pharmacology. Here we see different types of ion channels: voltage-gated, ligand-gated, and mechanically gated ion channels interact to generate and regulate neuronal communication. Voltage-gated channels control the action potential's rise and fall. Ligand-gated channels mediate synaptic signaling, and inhibitory channels maintain the balance necessary for stability. Ion channels are more than molecular gates. They are the electrical foundation of life. They convert chemical gradients into electrical signals that coordinate every heartbeat, reflex, and thought. When they malfunction, as we've seen with channelopathies, the consequences can be devastating. Understanding these processes at the molecular level not only enriches our grasp of physiology, but also informs medical innovation. Modern medicine continues to build upon the discoveries of channel physiology. As we move into future research, the study of ion channels remains central to understanding the brain's electrical symphony. Thank you.